Hello everyone, my name is Alexander Malinick, and today I'll be presenting my research on surface plasmon resonance imaging in combination with machine learning for microanalysis of multiple sclerosis biomarkers in whole serum. So multiple sclerosis is the most common autoimmune disease affecting young adults. And while there are various ways to detect and monitor this disease progression, a lot of them are limited and are considered indirect and unreliable, which has led for a great deal of interest to monitor biomarkers for this disease in serum, specifically anti-ganglioside antibodies, and gangliosides are important components of the myelin sheath, which causes the symptoms associated with multiple sclerosis as the myelin sheath is damaged. Some antibodies of interest are anti-GT1B, which is associated with loss of muscle control, anti-GM1 and anti-GA1, which result in changes in loss of vision. So we detect these antibodies using a PFD-TS surface, and then we incubate gangliosides onto them, which self-insert into that PFD-TS surface, allowing for the antigenic binding site to be displayed in a well-organized and easy-to-access manner. So this would be the ganglioside, and this would be the PFD-TS surface. And then we can capture and detect the antibodies of interest, allowing for us to detect and monitor the disease's progression. But we need to make sure our surface can actually detect and capture these antibodies. So the first step is we saturate the surface with 10% serum, which reduces the amount of non-specific and cross-reactivity when we introduce the spiked whole serum samples. As you can see here, there's a massive shift due to the amount of serum, but once we introduce the rinse period, we get that specific antibody-antigen interaction. But we investigate that with the different investigated antibodies, on each one of their ganglioside surfaces. And you can see here there is a difference in after the rinse cycle. Now, we also use endpoint data, which is the subtraction of post-rinse versus pre-rinse, and you can see that the specific antibody-antigen interactions overshadow everything else. But there is notable cross-reactivity between anti-GA1 and anti-GM1 in the respective surfaces. But there's little to no cross-reactivity between anti-GT1B and anti-GA1 and their respective surfaces. That has to do with the number of sialic acids. GT1B has 3 sialic acids, GM1 has 1 sialic acid, and GA1 has no sialic acids. But we want to be able to detect these antibodies at biological relevant concentrations. So we did a calibration curve and did detection of various concentrations. And these antibodies appear between 3 to 20 nanograms per ml, and we're able to detect within that range. So that shows that this could be used as a way to monitor and detect multiple sclerosis. But detecting in serum is very complex. There's a lot of cross-reactive interactions that make data analysis very difficult. So to address that, we turned towards machine learning. Specifically, I'll talk about neural networks, which is one of several algorithms that we used. But this allows us to look at essentially these interactions with an algorithm that is trained with 70% of the collected data and tested with 30%. It works very similar to how our neural networks work, where we have an input of data, which in this case is the unknown antibody ganglioside interaction, then a hidden layer where we have computations, and then a base layer, the output layer where we get everything sent out and we determine what those interactions are. So for example, we looked at 3 antibodies at 50 nanograms per ml on a GT1B ganglioside surface. You can see it has 83% is 83% accurate in differentiating which antibody-antigen interactions are occurring. We did the same thing with 10 nanograms per ml of anti-GT1B on 3 different ganglioside surfaces, and this had an 88% accuracy of differentiating whether it was GT1B, GM1, or GA1 surfaces interacting with that anti-GT1B. And then we took all of that data and we looked at the interactions between the investigated and the light antigens. And we found that with looking at cross-reactivity and that specific antibody-antigen interaction, we had a 96% accuracy of differentiating them based upon endpoint data and which interactions are actually occurring. So this shows that we have developed a platform capable of screening and differentiating between antibodies and serum at disease-relevant concentrations, which may drastically reduce the time and complexity of achieving an accurate diagnosis for various autoimmune diseases. We've also applied robust statistical analysis and machine learning to surface plasmon resonance imaging, allowing for a more comprehensive investigation into antibody-antigen interactions than what traditional endpoint assays can achieve. I'd like to say a great thank you to those who have sponsored this research, as well as my lab, who have been a great support. And if you'd like to talk to me more about my research, please feel free to message me and add me on LinkedIn. Also, a great thank you to the sponsors for this event that have allowed this to occur. And thank you to all of you who have put this together and who are watching my video. I really look forward to hopefully hearing from you all. Thank you so much and goodbye.